Okay, let's really unpack this. What truly makes you you? And I don't just mean, eye color or height. I mean the deep stuff, the fundamental, almost mind-boggling complexity happening inside every single one of your cells right now, this very second. It's like this constant, intricate choreography.
It really is. And at the absolute heart of that choreography, calling all the steps, you've got your genes. They are the fundamental control system for life. I mean, they dictate everything, how your cells get energy, how they build themselves, how they reproduce.
Now they even know what kind of cell to be, right? Like
Yeah.
a brain cell versus a muscle cell.
Exactly. And unfortunately, also what happens when that incredible system breaks down, which can lead to things like cancer.
So this deep dive is all about getting right to the core of that system. We're working from some really detailed source material on genetic control, the kind of stuff that really lays out the core mechanics, for anyone who needs that solid professional level grasp.
Yeah, our mission today is to take this incredibly intricate genetic blueprint and unpack it piece by piece, layer by layer. We want you to walk away with a really solid understanding of how it's built, how it stores all that crucial information, how that information gets read and acted upon, basically why it's so essential to everything your body does.
Okay, let's dive in. The master controllers themselves, genes. Where are they, and what exactly are they controlling on a daily basis?
Right. So genes live primarily inside the nucleus of, well, pretty much every cell in your body. And their control is really twofold. Yes, they determine heredity, the traits you inherit from your parents. But just as critically, they exert moment-to-moment control over the daily function of every single cell.
So they're not just about family history, they're like the operating manual for right now. How do they actually wield that control?
They do it by holding the instructions for building almost everything the cell needs. Specifically, they determine which structures, which enzymes, which chemicals get synthesized. If you picture it visually, it's often shown as the central dogma of molecular biology. DNA makes RNA, and RNA makes protein.
Ah, yeah, the classic flow. DNA to RNA to protein.
Exactly. The genetic code, which is locked away safely in the DNA inside the nucleus, first gets copied or transcribed into an intermediary molecule, RNA. This RNA molecule then travels out of the nucleus into the main body of the cell, the cytoplasm. And there, its message is translated into a specific protein molecule. That whole sequence DNA instruction in the nucleus, RNA messenger, final protein product in the cytoplasm, that's gene expression.
And how many of these genes, these instruction sets, are we talking about in a typical human cell?
It's estimated there are roughly 20,000 to 25,000 different genes in the human genome that actually code for proteins. But here's where it gets really interesting and adds a layer of complexity. The RNA that's copied from a single gene isn't always processed in the exact same way.
What do you mean processed?
Well, during a step called splicing, different segments of the initial RNA molecule can be cut out or kept. Think of it like editing a film. You can create different versions from the same raw footage.
Wait, so one gene can actually lead to different versions of a protein?
Precisely. This alternative splicing is a key reason why, even with only about 25,000 protein coding genes, our bodies can produce a vastly larger number estimated at over 100,000 different proteins.
Wow. Okay, that explains a lot more variety. So what are all these proteins actually doing in the cell?
They're basically the cell's workforce and its building materials. Some proteins are structural. They form the cell's internal skeleton, organelles, membranes, often working together with lipids and carbohydrates. But the vast, vast majority of proteins are enzymes.
Enzymes, the catalysts.
Exactly. Enzymes are the master catalysts. They speed up and control almost all of the thousands upon thousands of chemical reactions that keep the cell alive, generating energy, building molecules, breaking things down.
So the genes hold the blueprints for the enzymes, which then do all the work. It's like the system builds its own tools.
Okay, let's zoom right in on the blueprint itself, the DNA molecule. What does this incredibly important molecule actually look like? What's it made of?
Well, DNA molecules are these incredibly long, double-stranded structures. They're twisted into that famous shape, the double helix, like a spiral staircase. They reside in the cell nucleus, linked together end-to-end within the chromosomes, and they're huge molecules, molecular weights in the billions.
Okay, a twisted ladder. What makes up the sides of the ladder, and what are the rungs made of?
The sides, or the backbones, are formed by alternating units of phosphoric acid and a specific type of sugar called deoxyribose. They link together to form these two long strands. The rungs that connect the two backbones are formed by pairs of nitrogenous bases.
Nitrogenous bases. Those
Yeah.
are the A, G, C, T things.
Just them. There are four types. Two are purines, adenine, which we call A, and guanine, G. And two are pyrimidines, thymine, T, and cytosine, C. And when you put together one phosphoric acid, one deoxyribose sugar, and one of those four bases, A, G, C, or T, that gives you the fundamental building block, a nucleotide.
That's exactly right. A nucleotide is like one complete unit, the sugar, the phosphate group, and one base attached. So you have four types of DNA nucleotides corresponding to the four bases. Deoxydenilic acid with A, deoxythymidelic acid with P, deoxyguanilic acid with G, and deoxycytidilic acid with C.
Okay, so the two long strands are just chains of these nucleotides linked together. How do those two strands actually connect to form the double helix?
They're held together by chemical bonds specifically, weak hydrogen bonds that form between the bases on the opposite strands. These base pairs form the rungs of the ladder. And here's the absolutely critical rule, the foundation of the genetic code. The pairing is highly specific. Adenine, A, on one strand, always pairs up with thymine, T, on the opposite strand. It's always an AT pair. And guanine, G, on one strand, always pairs up with cytosine, C, on the other. Always a GC pair.
A with T, G with C, always. Is that specific pairing why DNA is so good at storing information and being copied accurately?
Absolutely. That complementary base pairing is fundamental. It means the sequence of bases along one strand automatically dictates the sequence on the other strand. If you know one, you know the other. And because those hydrogen bonds holding the pairs together are relatively weak, the two strands can be pulled apart fairly easily when the cell needs to read the information or make a copy. That's essential.
And the twist itself, the helical structure, is there a typical spacing?
Yeah. Typically, there are about 10 of these base pairs, 10 rungs on the ladder for every complete turn of the double helix.
Okay, got the structure. Double helix, nucleotide building blocks, sugar phosphate backbones, and crucial ATGC base pairing holding it together. Now, how is the actual information, the genetic code, stored within that structure?
The information, the genetic code itself, is encoded in the linear sequence of those bases A, G, C, and T along just one of the DNA strands. When the two strands temporarily separate for processes like reading the gene, the sequence of exposed bases on one strand acts as the instruction. And the code isn't read one base at a time, but in groups of three consecutive bases. Each three-base unit is effectively a code word, technically called a triplet.
So not just AGCT, but sequences like, say, AGC or CTT or GGC.
Exactly. And each of these three-base triplets specifies a particular amino acid. Remember, amino acids are the building blocks that make up proteins. For instance, the DNA triplet GGC is the code word for the amino acid proline. The triplet AGA codes for serine, and CTT codes for glutamic acid. So the specific sequence of these triplets along the DNA gene dictates the precise sequence of amino acids that will be assembled to build a specific protein.
That's the blueprint. 3 DNA letters equal one specific amino acid instruction. But you said the DNA stays safe in the nucleus and the proteins get built out in the cytoplasm. How does the message actually get from the DNA to the construction site?
Right, that's where RNA comes in as the essential intermediary. The process of copying the DNA code into RNA is called transcription. Because the master DNA blueprint can't leave the nucleus, the cell makes a portable copy of the specific gene's instructions onto an RNA molecule. This RNA molecule is synthesized right there in the nucleus, using the DNA gene as a template. Then this messenger RNA travels out through tiny pores in the nuclear membrane into the cytoplasm, carrying the code to where the proteins are actually assembled.
Okay, transcription making the RNA copy. How does the cell physically make that RNA molecule? What are its building blocks?
RNA building blocks, the RNA nucleotides, are very similar to DNA nucleotides, but with two key differences. First, the sugar in RNA is ribose, not deoxyribose like in DNA. It has one extra oxygen atom. Second, instead of the base thymine, T, RNA uses a different base called uracil. So the four bases in RNA are adenine A, guanine G, cytosine C, and uracil U.
So RNA nucleotides have ribose sugar and use U instead of T.
Got it. How are these RNA nucleotides actually assembled into a chain using the DNA template? The whole process is orchestrated by a crucial enzyme called RNA polymerase. First, the section of the DNA double helix containing the gene unwinds, and the two strands separate just in that region. The RNA polymerase enzyme then recognizes and binds to a specific start signal sequence on the DNA called the promoter, which is located just upstream of the gene itself. Once bound, RNA polymerase starts moving along one of the DNA strands, the template strand. As it moves, it reads the DNA bases and adds the corresponding complementary RNA nucleotides one by one, linking them together to form the growing RNA chain.
And the pairing rules are slightly different now because RNA has uracil instead of thymine?
Exactly right. The complementarity rule is adjusted. Where the DNA template strand has a guanine G, RNA polymerase adds a cytosine C to the RNA chain. Where DNA has cytosine C, RNA gets a guanine G. Where DNA has thymine T, RNA gets an adenine A. And this is the key difference where DNA has adenine A, RNA polymerase adds a uracil U.
Okay, so DNA's A pairs with RNA's U.
Precisely. The polymerase uses energy, actually derived initially from ATP, to activate the RNA nucleotides, and then uses that energy to form the bonds, linking them into the chain. It continues moving along the DNA, reading the template, and building the RNA copy until it reaches a specific stop signal sequence on the DNA. At that point, the RNA polymerase detaches. The newly synthesized RNA molecule is released, and the DNA strands zip back together again. The RNA molecule is now free in the nucleoplasm.
So the DNA's genetic message is transcribed into a complementary RNA sequence. You mentioned earlier there are different types of RNA involved. It's not just one messenger.
That's right. While ongoing research is revealing many types of RNA, especially non-coding ones with regulatory roles, our source focuses on 6 key types that are central to protein synthesis and its regulation.
Okay, let's run through them. First up, pre-mRNA.
Precursor messenger RNA, or pre-mRNA, is the very first raw RNA molecule that's transcribed directly from the DNA template in the nucleus. It's typically quite large and immature. Critically, it contains non-coding sequences called introns, which are interspersed between the actual coding sequences called exons. These introns need to be removed.
And that removal process, splicing, involves another type of RNA.
Yes, that's the job of small nuclear RNA, or snRNA. These snRNAs combine with proteins to form complexes called spliceosomes, which are the molecular machines that recognize the boundaries between introns and exons, cut out the introns, and splice the exons together, producing the mature mRNA.
Okay, so pre-mRNA gets processed, introns out, exons join to become the actual messenger.
Exactly. That final process molecule is the messenger RNA, mRNA. This is the molecule that carries the actual genetic code, the sequence of instructions for building a protein from the nucleus out to the cytoplasm. It's typically a long, single strand composed of hundreds or thousands of nucleotides. It contains a series of 3 base code words, now called codons, which are complementary to the triplets on the original DNA gene.
So if the DNA had GGC, AGA, CTT, the mRNA would have the codons CCG, UCU, GAA, carrying the instructions for proline, serine, and glutamic acid.
Precisely. The sequence of codons on the mRNA is the specific instruction manual read by the protein building machinery.
Okay, mRNA is the manual, but you need the actual building blocks, the amino acids brought to the site. Is that transfer RNA?
That's the role of transfer RNA, or tRNA. These are the delivery trucks. tRNA molecules are relatively small, usually around 80 nucleotides long. They fold up into a specific, complex, 3-dimensional structure, often depicted in 2D as a cloverleaf shape. The key thing is that each type of tRNA molecule is specifically designed to recognize and attach to one particular type of amino acid.
So there's a specific tRNA for proline, a different one just for serine, another just for glutamic acid, and so on for all 20 common amino acids.
Correct. And crucially, each tRNA molecule also has a special 3-base sequence located in a loop region called the anticodon. This anticodon is complementary to a specific mRNA codon. For example, the tRNA carrying proline would have an anticodon, like GGC, that can base pair with the mRNA codon for proline, CCG. When the tRNA arrives at the protein synthesis site, its anticodon temporarily binds to the matching codon on the mRNA. This ensures that the correct amino acid is delivered to the right place in the growing protein chain, following the instructions on the mRNA.
Okay, mRNA has the instructions, tRNA brings the right amino acids. Where does the actual construction happen? What reads the manual and links the amino acids together?
That's the job of the ribosome. The ribosome is essentially the cell's protein synthesis factory. And a major structural and functional component of the ribosome is ribosomal RNA, or rRNA. RNA molecules combined with numerous ribosomal proteins form the 2 subunits of the ribosome. These subunits come together on the mRNA molecule to form the functional ribosome. The ribosome is the physical platform where mRNA codons are read, tRNA anticodons bind, and the amino acids carried by the tRNAs are linked together in the correct sequence to form the protein.
So RNA is part of the factory machinery itself. Where are ribosomes made?
Ribosomal RNA is transcribed from specific genes in the DNA and processed within a specialized region of the nucleus called the nucleolus. There, the RNA combines with ribosomal proteins, which are synthesized in the cytoplasm and imported back into the nucleus, to form the large and small ribosomal subunits. These subunits are then exported out to the cytoplasm. They only assemble into a fully functional ribosome when they bind to an mRNA molecule to start protein synthesis.
Right, okay. So those are the main players in actually building the proteins. Pre-mRNA, snRNA, mRNA, tRNA, and rRNA. What about the other two mentioned microRNAs and small interfering RNAs? They sound like regulators.
They absolutely are regulators. These fall under the umbrella of non-coding RNAs. They don't code for proteins themselves, but they play critical roles in controlling gene expression, often by interfering with the mRNA messages. MicroRNAs, miRNAs, are very short, single-stranded RNA molecules, typically just 21 to 23 nucleotides long. They're processed from longer precursor RNAs transcribed from specific miRNA genes. After processing in the nucleus and cytoplasm, the mature miRNA gets incorporated into a protein complex called the RNA-induced silencing complex, or RISC.
RSC silencing complex. How does it silence genes?
The miRNA within the RISC complex acts as a guide. It directs RISC to bind to specific mRNA molecules that have sequences complementary to the miRNA. Once bound, the miRNA RISC complex can interfere with that target mRNA in a couple of ways. It can block the ribosome from translating the mRNA into protein, effectively stopping production. Or it can trigger the degradation, the destruction of the mRNA molecule itself. So miRNAs act like fine-tuners or off-switches for the expression of specific genes acting after the mRNA has been made. They're crucial for normal development and cell function, and problems with miRNA regulation are linked to various diseases, including cancer and heart disease.
Tiny molecules with big regulatory power. What about the siRNAs? Small interfering RNAs.
Small interfering RNAs, siRNAs, are functionally very similar to miRNAs. They are also short RNA molecules, usually 20-25 nucleotides, and they also get incorporated into the RISC complex to target specific mRNAs for silencing or degradation. A key difference is that siRNAs are often derived from double-stranded RNA precursors. In research and potentially therapeutic settings, siRNAs are frequently synthesized artificially to have sequences perfectly complementary to a target mRNA of interest.
So researchers can design siRNAs to specifically shut down almost any gene they want to study.
Exactly. Because you can design insert A to match virtually any known mRNA sequence, they become incredibly powerful tools for experimentally silencing specific genes to figure out their function. They also hold great therapeutic potential. The idea is you can design siRNAs to target and shut down the expression of genes that are causing disease, for example, an overactive oncogene in cancer.
Fascinating. The potential there is huge. Okay, so we've followed the genetic information from the DNA blueprint through transcription into various forms of RNA. Now let's put it all together at the factory, the ribosome. This is the process of translation.
Translation is exactly that, translating the language of nucleotides, the mRNA codons, into the language of amino acids, the protein sequence. This whole process happens on the ribosomes out in the cytoplasm. An mRNA molecule first binds to a ribosome. The ribosome then starts to move along the mRNA strand, usually beginning at a specific start codon sequence, often AUG. As the ribosome travels along the mRNA, it reads the codons one by one in sequence.
Like a little machine scanning a barcode or reading ticker tape?
That's a great analogy. As each 3-base codon enters the reading frame of the ribosome, the specific tRNA molecule whose anticodon is complementary to that codon comes in and binds temporarily. This tRNA is carrying its specific amino acid. The ribosome then catalyzes the formation of a peptide bond between the newly arrived amino acid and the growing end of the polypeptide protein chain. The ribosome then shifts one codon further down the mRNA. The empty tRNA, which has delivered its amino acid, is released, and the next codon is ready to be read, allowing the next correct tRNA to bind.
And the ribosome just keeps chugging along the mRNA, reading codons, bringing in tRNAs, and linking amino acids.
Yes, codon by codon, amino acid by amino acid, the protein chain gets longer and longer, assembled in the precise sequence specified of the mRNA codons. This elongation process continues until the ribosome encounters one of 3 specific stop codons on the mRNA. These codons don't specify an amino acid. Instead, they signal the end of the protein coding sequence. When a stop codon is reached, release factor proteins bind, the completed polypeptide chain is released from the ribosome, and the ribosome subunits detach from the mRNA, ready to start the process again on another mRNA molecule.
Can one mRNA molecule be used to make multiple protein copies simultaneously? It seems inefficient to just make one at a time.
Absolutely, it's much more efficient than that. As soon as the first ribosome is moved a bit down the mRNA from the start codon, a second ribosome can bind to the start site and begin translation as well. You often see multiple ribosomes, sometimes 3 to 10, even more, translating the same mRNA molecule at the same time. This structure, an mRNA with multiple ribosomes attached and translating, is called a polyribosome, or polysome. Each ribosome on the polysome is synthesizing a copy of the same protein, but they're all at different stages of completion, like an assembly line producing multiple products simultaneously from the same blueprint.
Makes sense for efficiency. Are all ribosomes just floating free in the cytoplasm or do some work in specific locations? I remember seeing images where they look attached to membranes.
That's a key point. While many ribosomes are free in the cytosol, synthesizing proteins that will function there, like many enzymes or structural proteins, a significant number become attached to the membrane of the endoplasmic reticulum, ER. This happens for proteins that are destined for secretion out of the cell, or insertion into cell membranes, or delivery to certain organelles like lysosomes. These proteins typically have a special signal sequence of amino acids at their beginning. As this sequence emerges from the ribosome during translation, it's recognized by a signal recognition particle, which guides the entire ribosome mRNA complex to dock onto specific receptor proteins on the ER membrane. As translation continues, the growing polypeptide chain is threaded through a channel in the ER membrane directly into the lumen, the space inside the ER, or inserted into the ER membrane itself. This association of ribosomes gives parts of the ER its characteristic rough or granular appearance in electron micrographs, hence the rough ER.
Ah, so the protein's destination can actually determine where it gets synthesized free ribosome or ER-bound ribosome. Okay, let's dig a bit into the actual chemistry now. Linking those amino acids together sounds like it must require energy.
Oh, it's remarkably energy intensive. Protein synthesis is one of the most energy-consuming processes in the cell. Let's break down the key chemical events. First, before a tRNA can even pick up its amino acid, that amino acid needs to be activated. This step uses the cell's primary energy currency, ATP, adenosine triphosphate. An enzyme links the amino acid to AMP, adenosine monophosphate, releasing two high-energy phosphate groups from the ATP. This forms an activated amino acid AMP complex, essentially priming the amino acid with stored energy.
So ATP gives the amino acid an energy boost to get it ready.
Exactly. Then this energized activated amino acid, still bound to AMP, reacts with its specific tRNA molecule. The energy stored in that amino acid AMP bond is used to covalently link the amino acid to the correct tRNA molecule, releasing the AMP. Now you have the charged amino acid tRNA complex ready for the ribosome.
Okay, amino acid activated, attached to its specific tRNA. Then it goes to the ribosome, matches its anticodon to the mRNA codon. How is the actual peptide bond formed between amino acids?
Within the large ribosomal subunit, there's an enzymatic activity attributed to the RNA itself called peptidyltransferase. This activity catalyzes the formation of the peptide bond. It essentially transfers the growing polypeptide chain from the tRNA sitting in one side of the ribosome onto the amino group of the new amino acid attached to the tRNA that just arrived in the adjacent site.
And forming that peptide bond, the link between amino acids, does that cost additional energy right there at the ribosome?
Yes, and this is where the really significant energy cost comes in. Beyond the initial ATP used for activation, the process of bringing the charged tRNA to the ribosome and forming the peptide bond requires the energy equivalent of breaking two more high-energy phosphate bonds, usually derived from another energy-carrying molecule called GTP, guanosine triphosphate. So if you add it all up, the initial activation, equivalent to two high-energy bonds from ATP breaking down to AMP, plus the steps on the ribosome, equivalent to another two high-energy bonds from GTP, the total energy cost to add just one single amino acid to a growing protein chain is the equivalent of breaking four high-energy phosphate bonds.
4 high-energy bonds per amino acid. That is a huge energy investment.
It really is. It underscores just how fundamentally important accurate protein synthesis is to the cell that it's willing to expend so much energy on it.
Wow. Okay, so we have all these proteins being made, structural ones, thousands of enzymes, all costing a lot of energy. Cells can't just be making everything all the time at full tilt. There must be incredibly precise control over which genes are turned on, when, and how much protein gets made.
Absolutely critical. You hit the nail on the head. Without powerful, intricate control systems, cells would waste enormous energy. Chemical reactions could run rampant, or cells could grow uncontrollably. Nature has evolved complex feedback mechanisms, often linked directly to the genes themselves or the enzymes they produce, to regulate these processes tightly.
And the source material points to 2 main strategies or levels of control.
Yes, we can broadly categorize cellular control into 2 main types: genetic regulation and enzyme regulation. Genetic regulation controls the very first steps. The degree of gene activation and the rate at which the gene's products, initially RNA, ultimately protein, are formed. This encompasses control over transcription and translation. This level of control is absolutely essential for cells to respond to their environment, and crucially, for cell differentiation, it's how a heart muscle cell ends up functioning so differently from, say, a kidney cell, even though they contain the same basic set of genes. The ultimate measure here is how much functional protein is produced from a specific gene.
And the second type, enzyme regulation.
Enzyme regulation deals with controlling the activity level of enzymes that have already been synthesized. So even after the protein, the enzyme, is made, the cell can still turn its activity up or down or switch it on or off based on current needs.
Okay, let's tackle genetic regulation first, controlling if and how much protein gets made from the gene blueprint. Where does this control typically start?
It largely begins right at the source. Controlling the initiation and rate of transcription, the process of making RNA from the DNA template. The key control element on the DNA molecule itself, right near the gene, is called the promoter.
Promoter, that's the landing site for the RNA polymerase enzyme to start copying the gene.
Exactly.
Ugh.
In eukaryotes, like human cells, the promoter region is complex. There's a core part called the basal promoter, located very close to the gene's starting point. This basal promoter often contains characteristic DNA sequences, like the famous TATA box, a sequence rich in TNA bases like TATA-A. This TATA box is a crucial binding site for general transcription factors, which are proteins required to recruit the RNA polymerase enzyme, specifically RNA polymerase II for protein-coding genes, and position it correctly to start transcription. The basal promoter is essential. Transcription simply can't begin without this core machinery assembling here.
So the basal promoter is like the essential on-switch platform, but genetic regulation is also about fine-tuning, making more or less protein, or only making it in certain cell types. Where does that control come from?
That comes from other regulatory DNA sequences, often located in the upstream promoter region, further away from the gene's start site, and also from elements called enhancers and silencers, which can be quite distant. These regions contain binding sites for a whole host of specific transcription factors. These are proteins that can either activate transcription, making it happen more frequently or efficiently, or repress transcription, slowing it down or shutting it off. The specific combination of these binding sites and the transcription factors available in a particular cell type determines how strongly and under what conditions a gene is expressed. It's this combinatorial control that allows for the vast differences in gene expression between different tissues and in response to different signals. These factors often influence the assembly or activity of the basal transcription machinery at the core promoter.
Okay, so the upstream promoter and its specific factors act like the dimmer switch or volume control. You mentioned enhancers can be far away. How do they influence a gene from a distance?
Enhancers are fascinating. They are DNA sequences that can bind activator proteins and significantly boost the rate of transcription of a target gene. What's remarkable is their location independence. An enhancer controlling a particular gene might be located thousands, even hundreds of thousands, of base pairs away, either upstream or downstream of the gene, sometimes even within an intron of the gene itself, or occasionally on a completely different chromosome.
How on earth can something so far away physically affect the promoter?
The key is the three-dimensional organization of DNA within the nucleus. DNA isn't just a long straight line. It's looped and folded and coiled extensively. This looping can bring regions that are linearly distant on the DNA molecule into close physical proximity. So, an enhancer sequence, with its bound activator proteins, can loop around and make direct contact with the transcription machinery assembled at the promoter of its target gene, stimulating its activity. The human genome is thought to contain hundreds of thousands of these enhancer elements, adding incredible complexity to gene regulation.
Wow, a whole network of long-distance remote controls. And what about insulators? They sound like they do the opposite.
Chromosomal insulators, or boundary elements, act like fences. They are DNA sequences that help to partition the genome into functionally independent domains. They essentially block the influence of regulatory elements, like enhancers or silencers, from spreading inappropriately to neighboring genes. So an enhancer meant for gene A doesn't accidentally activate gene B next door if there's an insulator between them. Insulator activity itself can be regulated, for example, by chemical modifications to the DNA like methylation. They help maintain the specificity of gene regulation in complex genomes.
So they maintain regulatory neighborhoods, preventing crosstalk. Are there other ways genetic regulation happens beyond these DNA elements?
Well, yes. There are regulatory genes that produce proteins which themselves act as transcription factors, activators or repressors, for other genes. Control can also occur after transcription is started. We already mentioned alternative splicing of pre-mRNA as a way to generate different proteins from one gene. There's also control over mRNA stability, how long the mRNA molecule lasts in the cytoplasm before being degraded, and control over the efficiency of translation, how readily ribosomes bind and translate the mRNA.
And doesn't the way the DNA is packaged itself play a role? You mentioned it's wound around proteins, histones.
Absolutely crucial. This is often called epigenetic regulation. The DNA in our chromosomes is wrapped around spool-like proteins called histones. These DNA histone units, called nucleosomes, are then further compacted and folded. When DNA is very tightly packaged in this condensed chromatin structure, it's physically inaccessible to the transcription factors and RNA polymerase. The genes in that region are effectively silenced.
So the cell can physically lock away sections of the genome it doesn't need.
Exactly. This is a fundamental level of large-scale gene control. Conversely, for a gene to be transcribed, the chromatin structure around it must be loosened or opened up. There are enzymes that chemically modify histones, adding or removing things like acetyl or methyl groups, or remodel the chromatin structure, making the DNA more accessible. Regulatory proteins and external signals like hormones can trigger these modifications, leading to the activation or repression of specific genes or entire chromosomal regions. It's an incredibly dynamic and complex system.
It really sounds like multiple layers of control operating simultaneously. Okay, that covers genetic regulation controlling if and how much protein is made. Let's switch to the second type, enzyme regulation. How does the cell control the activity of enzymes that are already present?
This level of control is often faster, acting directly on the enzyme molecules themselves. It involves intracellular chemical substances that can either inhibit or activate specific enzymes. A very common mechanism is enzyme inhibition, particularly feedback inhibition. Imagine a metabolic pathway where substance A is converted to B by enzyme 1, B to C by enzyme 2, and C to D by enzyme 3. The final product, D, often acts as an inhibitor of the first enzyme in the pathway, enzyme 1.
So, if the cell accumulates enough of the final product, D, that product itself goes back and tells the beginning of the assembly line to slow down or stop.
Precisely. This inhibition usually occurs through allosteric regulation, where the end product, D, binds to enzyme 1 at a site different from the active site where substance A binds. This binding changes the enzyme's shape, making it less effective or completely inactive. It's a classic negative feedback loop. It prevents the cell from wasting energy making too much of product D, and also prevents the buildup of intermediate products, B and C. This is how the synthesis of many essential molecules like amino acids and nucleotides is regulated.
Makes perfect sense. And the opposite can happen too, enzyme activation.
Yes, enzyme activation is also common. Sometimes enzymes are synthesized in an inactive form, a proenzyme or zymogen, and need to be activated only when needed. Other times, the activity of an existing enzyme is boosted by an activator molecule. A classic example involves energy metabolism. When ATP levels in the cell get low, levels of cyclic AMP, CMP, often rise. CMP then acts as an allosteric activator for certain enzymes, like phosphorylase, which breaks down stored glycogen into glucose. This glucose can then be used to generate more ATP, restoring the cell's energy balance. CMP here is the activator signal.
A quick response mechanism to changing needs. The source also mentioned an interesting cross-regulation example between purines and pyrimidines.
Yes, that's a beautiful example of sophisticated enzyme regulation ensuring balance. Purines, A, G, and pyrimidines, CTU, are the building blocks for DNA and RNA, and they're needed in roughly equal amounts. The metabolic pathways that synthesize purines are typically inhibited by purines themselves, feedback inhibition, but they are activated by pyrimidines. Conversely, the pathways synthesizing pyridines are inhibited by pyridines but activated by purines. This intricate crosstalk ensures that the cell maintains a balanced supply of both types of building blocks, which is absolutely critical for accurate DNA replication and RNA synthesis.
That's really elegant. These interlocking controls, genetic and enzymatic, are constantly working to keep everything running smoothly inside the cell. And this level of control is absolutely fundamental when we think about how cells reproduce, which is really the core of life itself.
It truly is. The entire DNA genetic system, with all its regulatory layers, ultimately controls not just the cell's moment-to-moment function and growth characteristics, but also the critical decision of when and if a cell will divide and reproduce. This control over the cell cycle is central to everything from the development of a multicellular organism, from a single egg, to the maintenance of tissues in an adult.
Okay, the cell cycle. The definition is the period from one cell reproduction event to the next. How long does this cycle typically take?
It varies enormously depending on the cell type and the conditions. For rapidly dividing cells, like some stem cells in bone marrow or the cells lining your gut, the entire cycle might take only 10 to 30 hours. Interestingly, the actual process of division, mitosis, is usually quite short, often only about 30 minutes to an hour.
So most of the cell's life in the cycle isn't spent actively dividing.
Not at all.
Yeah.
Over 95% of the cell cycle duration for dividing cells is spent in interphase. This is the period between mitotic divisions. Interphase is a busy time, though. The cell is actively growing, carrying out its specialized functions, and, critically, preparing for the next division. The most important preparatory event during interphase is the replication of its DNA. Preliminary changes needed for mitosis also begin late in interphase.
And not all cells are constantly cycling, right? Many cells in our body seem to stop dividing.
Absolutely. The cell cycle is tightly regulated, and various inhibitory factors can slow it down or stop it altogether. Many differentiated cells in adults enter a quiescent state called G0, where they are not actively preparing to divide. Some cells, like mature nerve cells and skeletal muscle cells, generally lose the ability to divide after they differentiate during development and remain in this non-dividing state for the rest of their lives. Others, like liver cells, normally don't divide much, but can re-enter the cell cycle and proliferate rapidly if the liver is damaged. And then you have cells like those in the bone marrow or skin epidermis that are cycling and dividing continuously throughout life.
So, huge variation. But for any cell that is going to divide, the absolute prerequisite is copying its DNA blueprint. That's DNA replication.
Yes, DNA replication is the essential first step. Before a cell can undergo mitosis and divide into two daughter cells, it must accurately duplicate its entire genome, ensuring that each new cell receives a complete and identical set of genetic instructions.
When exactly does this replication happen during the cell cycle?
It occurs during a specific period within interphase, known as the S phase, S for synthesis. In a typical mammalian cell cycle, the S phase might start 5 to 10 hours before mitosis actually begins, and the replication process itself usually takes about 4 to 8 hours to complete.
Okay, S phase and interphase. How does DNA replication compare to transcription, where we saw RNA being made from a DNA template?
There are some similarities, but also crucial differences. Like transcription, replication uses the DNA strands as templates and follows base pairing rules. However, 1. In replication, both strands of the DNA double helix serve as templates simultaneously, whereas in transcription, typically only one strand is used as a template for a given gene. 2. Replication copies the entire length of the DNA molecule in each chromosome, end to end. Transcription copies only specific, relatively short segments, the genes. 3. The main enzymes are different. Replication relies heavily on enzymes called DNA polymerases to synthesize the new DNA strands, and DNA ligas to join DNA fragments together. Transcription uses RNA polymerase. 4. The end product is different. Replication produces two double-stranded DNA molecules, while transcription produces single-stranded RNA molecules.
Okay, key differences. How does the process actually start? How does the cell access the templates in the double helix?
The process begins at specific sites along the DNA called origins of replication. Here, initiator proteins bind, and an enzyme called DNA helicase starts to unwind the double helix, breaking the hydrogen bonds between the base pairs. This unwinding separates the two parental DNA strands, creating a Y-shaped structure known as the replication fork. This fork is the active site where replication occurs, and it moves along the DNA as unwinding proceeds. Eukaryotic chromosomes have multiple origins of replication, allowing the long DNA molecules to be copied more quickly.
And I remember DNA strands have directionality, a 5' end and a 3' end. Does that matter for replication?
It matters immensely. DNA polymerase, the enzyme that builds the new DNA strand, can only add new nucleotides to the 3' end of a growing strand. It can only synthesize DNA in the 5 to 3' direction. Because the two parental strands in the double helix run in opposite directions, anti-parallel, this directional constraint means the two new strands at the replication fork must be synthesized differently.
Custom.
One parental strand, oriented 3 beta 5 beta relative to the movement of the replication fork, can be used as a template for continuous synthesis of a new strand, called the leading strand, in the 5' to 3' direction, moving right along with the fork. However, the other parental strand is oriented 5' to 3' relative to the fork's movement. DNA polymerase cannot synthesize continuously on this template because that would require 3' to 5' synthesis. So this new strand, the lagging strand, must be synthesized discontinuously.
Discontinuously. How does that work?
The lagging strand is synthesized in a series of short fragments. As the fork opens up, an enzyme called DNA primase synthesizes a short RNA primer on the lagging strand template. DNA polymerase then starts from that primer and synthesizes a short DNA fragment called an Okazaki fragment. in the 5 to 3' direction, moving away from the replication fork until it hits the previous primer. As the fork unwinds further, a new primer is laid down further along the lagging strand template, and another Okazaki fragment is synthesized. So the lagging strand is built piece by piece, backwards relative to the overall direction of fork movement.
Okay, leading strand is continuous, lagging strand is made in these Okazaki fragments, starting from RNA primers. What happens to those primers and fragments?
Good question. First, the RNA primers need to be removed. This is done by other enzymes, often exonucleases. Then, a different type of DNA polymerase fills in the resulting gaps with the correct DNA nucleotides. Finally, the enzyme DNA ligase joins the sugar phosphate backbones of the Okazaki fragments together, creating a continuous, complete lagging strand.
It's quite a complex choreography on the lagging strand. Is there a system to check for errors during this whole process? Copying billions of bases seems prone to mistakes.
Absolutely, accuracy is paramount. DNA polymerases themselves have a proofreading ability. As they add a new nucleotide, they can check if it forms the correct base pair, AT or GC, with the template strand. If an incorrect nucleotide is added, the polymerase can often detect the mismatch, back up, remove the wrong nucleotide using an exon nucleus activity, and then insert the correct one before moving on. This proofreading significantly reduces the error rate. There are also other mismatch repair systems that scan the newly synthesized DNA shortly after replication and correct most of the errors missed by the polymerase's proofreading.
So multiple layers of error checking. What about the physical stress? Unwinding a long helix must create tension ahead of the fork.
It does. As the helicase unwinds the DNA at the fork, it causes the DNA ahead of the fork to become overwound or supercoiled. This torsional stress could potentially stall replication. To relieve this stress, enzymes called topoisomerases work ahead of the replication fork. They make temporary nicks or breaks in one or both DNA strands, allow the DNA to rotate and relax the supercoiling, and then reseal the breaks.
Amazing coordination. So, after all this unwinding, priming, synthesizing, proofreading, legating, and stress relief, what's the final result?
The final outcome is two complete double-stranded DNA molecules, each identical in sequence to the original parent DNA molecule. And importantly, each of these new DNA molecules consists of one strand from the original parent molecule and one newly synthesized strand. This mode of replication is called semi-conservative replication, because half of the original molecule is conserved in each of the two daughter molecules. These two new DNA double helices then coil up.
Semi-conservative replication, one old strand, one new strand in each copy. Okay, so the blueprint is duplicated. But despite all that proofreading and repair, do mistakes or mutations still sometimes slip through?
They do. While the overall fidelity of DNA replication or repair is incredibly high, it's not absolutely perfect. Occasionally an error is missed, or DNA gets damaged by other means like chemicals or radiation, and isn't repaired correctly before the next replication cycle. A permanent change in the DNA nucleotide sequence that results from these uncorrected errors or damage is called a mutation.
And what are the consequences if a mutation occurs in a gene?
It depends on the specific mutation and where it occurs. A mutation might change a codon, leading to the wrong amino acid being inserted into a protein, potentially altering its function. Or it might create a premature stop codon, resulting in a shortened, non-functional protein. Sometimes it might have no effect, a silent mutation, or it could even rarely be beneficial. But often, mutations in critical genes disrupt normal cell function, alter cell behavior, as in cancer, or can even be lethal to the cell or organism.
How often do these mutations realistically happen?
Even with a high accuracy, the sheer size of the genome means mutations do occur. It's estimated that perhaps 10 or more new mutations might arise spontaneously in the genome passed down from one human generation to the next. However, we have a built-in protection mechanism against the immediate effects of many mutations, diploidy. We inherit two sets of chromosomes, one from each parent. Most genes are therefore present in two copies. If one copy acquires a harmful mutation, the other, functional copy on the homologous chromosome can often compensate, preventing any obvious negative effect.
That redundancy is a huge advantage. Speaking of chromosomes, how does the cell package all of this replicated DNA? We're talking about meters of DNA per cell, fitting into a tiny nucleus.
It's an amazing packaging feat. The replicated DNA molecules don't just float around, they are meticulously organized and condensed into structures called chromosomes. Remember, human somatic cells typically have 46 chromosomes, arranged in 23 pairs. Each chromosome consists of a single, extremely long DNA double helix associated with a large amount of protein.
What kinds of proteins are involved in this packaging?
The main packaging proteins are histones. These are relatively small, positively charged proteins. The negatively charged DNA double helix wraps around cores of histone proteins, forming repeating units that look like beads on a string called nucleosomes. These nucleosomes are then further coiled and folded, with the help of other non-histone proteins, into progressively more compact structures, eventually forming the highly condensed chromosome visible during mitosis.
And you mentioned earlier, these histones aren't just passive spools, they also play a role in regulating gene activity.
Yes, that's a crucial point. The degree of DNA packaging is directly related to gene activity. When DNA is tightly wound around histones and compacted into dense chromatin, the genes in that region are generally inaccessible to the transcription machinery and are therefore turned off or silenced. Regulatory mechanisms can modify the histones or chromatin structure to loosen the packaging in specific regions, allowing the DNA to become accessible for transcription. So chromatin structure itself is a major layer of gene control.
So packaging equals control. What about the non-histone proteins?
There are many other types of proteins associated with chromosomes besides histones. These non-histone proteins have diverse roles. Some are structural, helping to organize the higher order folding of chromatin. Others are involved directly in genetic regulation, acting as transcription factors, activators, inhibitors, or enzymes that modify chromatin.
Okay, so after the DNA itself replicates during S phase, when do the chromosomes become fully duplicated structures?
Following DNA replication, the newly synthesized DNA molecules immediately associate with histone and non-histone proteins, assembling into two identical chromosome structures. These two identical copies, now called sister chromatids, remain physically attached to each other at a specialized region called the centromere. They stay joined like this until they are ready to be pulled apart during mitosis. So a chromosome entering mitosis actually consists of two identical sister chromatids.
Right, got it. DNA copied, packaged into duplicated chromosomes, sister chromatids held at the centromere. The cell is now fully prepped for the final act. Mitosis, the actual division into two cells.
Mitosis is the process where the nucleus divides, ensuring that each of the two new daughter cells receives one complete set of chromosomes, identical to the parent cell's set. In most cells, it follows fairly quickly after DNA replication is complete, maybe within an hour or two. It's a continuous process, but biologists divide it into distinct stages for ease of understanding.
Before the stages, the source mentions a mitotic apparatus forming. What is that composed of?
The formation of the mitotic apparatus is one of the first key events, starting in the cytoplasm during late interphase or early mitosis. Near the nucleus in animal cells are structures called centrosomes, each typically containing a pair of centrioles. These centrosomes also duplicate during interphase. As mitosis begins, the two centrosomes start to move apart, migrating through opposite poles ends of the cell. As they move, microtubules protein filaments begin to grow out from each centrosome. Some microtubules extend between the two separating centrosomes, forming the mitotic spindle. Other microtubules radiate outwards from each centrosome towards the cell periphery, forming structures called asters. The entire structure, the two centrosomes, the spindle microtubules, and the aster microtubules constitutes the mitotic apparatus. This apparatus is the framework that will orchestrate chromosome movement.
So the cell builds this elaborate scaffold to pull itself apart. Okay, let's walk through the stages, starting with prophase.
In prophase, several things happen concurrently. In the cytoplasm, the mitotic spindle continues to form and elongate as the centrosomes move further apart. Inside the nucleus, the replicated chromosomes, which were long and diffuse during interphase, begin to condense dramatically. They coil up tightly, becoming shorter and thicker, eventually becoming visible under a light microscope as distinct structures, each clearly consisting of two sister chromatids joined at the centromere.
Okay, chromosomes condense, spindle forms. Next stage, prometaphase.
Prometaphase begins abruptly when the nuclear envelope, the membrane surrounding the nucleus, breaks down in fragments. This allows the spindle microtubules, extending from the poles, to invade the nuclear area and make contact with the chromosomes. Specialized protein structures, called kinetochores, assemble on the centromeres of each sister chromatid. Specific spindle microtubules, now called kinetochore microtubules, attach to these kinetochores. These microtubules start exerting forces, jerking the chromosomes back and forth.
They're grabbing onto the chromosomes, then they line them up.
Yes, that alignment happens during metaphase. The push and pull forces exerted by the kinetochore microtubules attached to opposite poles eventually cause all the chromosomes to line up precisely along the midline of the cell. This imaginary plane is called the metaphase plate, located equidistant between the two spindle poles. At metaphase, the chromosomes are maximally condensed and clearly aligned.
Everything perfectly aligned at the equator. What triggers the separation in anaphase?
Anaphase is the shortest but most critical stage. It begins subtly when the proteins holding the sister chromatids together at the centromere are cleaved. The sister chromatids separate, and they are now considered individual daughter chromosomes. Immediately, the kinetochore microtubules begin to shorten, pulling the separated daughter chromosomes towards opposite poles of the cell. Simultaneously, the non-kinetochore spindle microtubules lengthen, pushing the poles further apart and elongating the entire cell. So one complete set of chromosomes moves to each pole.
Two identical sets of chromosomes moving to opposite ends. What happens in the final stage? Telophase.
In telophase, the separated daughter chromosomes arrive at the opposite poles of the elongated cell. The chromosomes begin to decondense, becoming less tightly coiled and reverting to their more extended interphase state. A new nuclear envelope begins to form around each set of chromosomes, using membrane vesicles derived from the parent cell's ER and nuclear envelope fragments. This creates two distinct daughter nuclei within the single elongated cell. The mitotic spindle disassembles. Mitosis, the division of the nucleus, is now essentially complete.
But the cell itself still needs to divide in two, right? The cytoplasm.
Yes, exactly. The division of the cytoplasm, called cytokinesis, usually begins during late anaphase, or telophase, and overlaps with the final stages of mitosis. In animal cells, a contractile ring, made primarily of actin filaments and myosin-motor proteins, forms just underneath the plasma membrane at the location of the old metaphase plate. This ring contracts, like pulling a drawstring, creating a groove called the cleavage furrow. The furrow deepens until it pinches the parent cell completely in two, resulting in two separate daughter cells, each with its own nucleus containing a complete set of chromosomes and roughly half of the cytoplasm and organelles from the parent cell.
And that completes the cell cycle, producing two genetically identical daughter cells. We talked earlier about how wildly cell division rates vary between different tissues. How is this growth and reproduction controlled at the level of the whole tissue or organism? It can't just be individual cells deciding.
That's right. The control of cell proliferation in a multicellular organism is incredibly complex and involves communication between cells. While it's not fully understood, several key mechanisms are known to play roles. As you noted, some tissues need constant renewal, like blood or skin. Others only divide to repair damage, like liver, and some hardly divide at all in adults, like neurons. This implies sophisticated control signals.
Is there some kind of sensing mechanism when more cells are needed, like after an injury?
Yes, compensatory growth is a clear example. If part of the liver is removed, the remaining cells rapidly divide until the liver regains its original size. This suggests signals are involved. Some known mechanisms include 1. Growth factors. These are signaling molecules, often proteins, that stimulate cell division. They can be secreted by nearby cells or circulate in the bloodstream. Different cell types respond to specific growth factors. For example, platelet-derived growth factor, PDGF, stimulates the division of fibroblasts during wound healing. 2. Contact inhibition. Many normal cells exhibit this property, especially when grown in culture. They will proliferate until they form a single continuous layer, a monolayer, and then they stop dividing once they make contact with their neighbors. This density-dependent inhibition likely plays a role in controlling tissue size and preventing overgrowth in the body.
They stop when they sense they're crowded.
Exactly. 3. Accumulation of secretions. Sometimes cells seem to stop growing when substances they themselves secrete build up in their environment. This could be a form of negative feedback, where the cell's own products signal that the population is dense enough.
Interesting checks and balances. Now let's shift to something you mentioned connects to both aging and cancer. Telomeres. What exactly are they?
Telomeres are specialized structures found at the very ends of linear eukaryotic chromosomes. They consist of long stretches of repetitive DNA sequences. In humans, the sequence TTG is repeated hundreds or thousands of times, and associated proteins. You can think of them as protective caps on the chromosome ends, like the plastic tips on shoelaces.
What are they protecting the chromosome ends from?
They primarily protect the vital gene-coding regions of the chromosome from being gradually eroded during successive rounds of DNA replication. There's a fundamental problem with replicating the very ends of linear DNA molecules. The DNA replication machinery requires an RNA primer to get started, and while primers within the strand can be removed and replaced with DNA, the primer at the absolute 5-foot end of a newly synthesized strand cannot be fully replaced once removed. This means that with each round of replication, a small segment of DNA is lost from the end of the chromosome.
So every time a cell divides, its chromosomes get slightly shorter at the ends.
Precisely. The telomeric repeats act as a disposable buffer zone. They contain no essential genetic information, So losing a small number of these repeats with each division doesn't harm the cell initially. It prevents the loss of actual genes located further inward on the chromosome.
How does this relate to aging?
This progressive telomere shortening is strongly linked to cellular aging or senescence. In most normal human somatic cells, telomeres lose anywhere from 30 to 200 base pairs of repetitive sequence with each cell division. After a certain number of divisions, known as the Hayflick limit, the telomeres become critically short. This triggers a DNA damage response, often leading the cell to permanently stop dividing, enter senescence, or to undergo programmed cell death, apoptosis. This limit on the number of times most cells can divide is thought to contribute to the aging process at the tissue and organism level. Erosion can be accelerated by factors like oxidative stress.
So telomere length acts like a cellular clock, counting down the number of divisions left.
In a sense, yes, for most normal cells. However, there's an enzyme called telomerase that can counteract this shortening. Telomerase is a specialized reverse transcriptase that carries its own RNA template and can add the repetitive TTG sequences back onto the ends of chromosomes. Telomerase is highly active in germ cells, which produce sperm and eggs, needing to maintain telomere length across generations, and in some stem cells, which need to divide extensively throughout life. It's also active during embryonic development.
But in most of our regular adult body cells, telomerase is switched off or present at very low levels.
Correct. The low or absent telomerase activity in most somatic cells is what leads to the progressive telomere shortening and the eventual limit on cell proliferation. This limitation is actually thought to be an important tumor suppressor mechanism.
Preventing cells from dividing indefinitely, which brings us straight to the cancer connection.
Exactly. One of the key characteristics or hallmarks of most cancer cells is their apparent immortality, their ability to divide essentially without limit, bypassing the normal senescence program. In a large majority of human cancers, estimated 85–90%, this is achieved through the reactivation or upregulation of telomerase. The cancer cells switch telomerase back on, allowing them to continuously rebuild their telomeres and thus avoid the telomere shortening crisis that would normally stop their division. Maintaining telomere length is crucial for the uncontrolled proliferation of cancer cells.
So a natural aging tumor suppressing mechanism is overcome by cancer cells hijacking telomerase. That's a profound link. Quickly then, how is the size of a cell generally controlled?
Cell size seems to be primarily determined by the amount of functional DNA in the nucleus, which in turn dictates the cell's capacity for synthesizing RNA and protein. Generally, a cell grows during interphase until it reaches a size appropriate for its DNA content before it divides. If you experimentally block mitosis, cell division, but allow DNA replication to continue, using a drug like colchicine, for example, the cell will end up with double the normal amount of DNA. Such cells typically grow to become proportionately larger, presumably because the increased DNA allows for greater rates of RNA and protein synthesis, supporting a larger cell volume.
More blueprint capacity allows for a bigger structure. Makes intuitive sense. Okay, let's move to one of the most fundamental processes in biology. How all the diverse, specialized cells in our body arise from a single starting cell. Cell differentiation.
Cell differentiation is the process by which cells become specialized in structure and function. It's how a generic embryonic cell gives rise to a neuron, a muscle cell, a skin cell, a liver cell, and all the other varied cell types that make up a complex organism.
The source mentions a classic frog experiment related to this. Does differentiation involve losing genes?
That experiment was key. Researchers took the nucleus from a fully differentiated cell, like an intestinal cell, of a tadpole, and transplanted it into a frog egg cell whose own nucleus had been removed. Remarkably, in many cases, this reconstructed egg could develop into a normal tadpole, and sometimes even a frog. This demonstrated that even a highly specialized differentiated cell still contains the complete set of genes required to build the entire organism. Differentiation doesn't involve throwing away genes, it involves selectively using them.
So if all differentiated cells, mostly, have the same genes, how do they become so different in function and form?
It's achieved through differential gene expression specifically, the stable and heritable repression of large sets of genes. As cells differentiate down specific lineages, certain groups of genes that won't be needed for that cell type's function are permanently silenced. The mechanism often involves changes in chromatin structure. Electron microscopy shows that certain segments of DNA associated with histones become highly condensed in differentiated cells, preventing them from being transcribed. The hypothesis is that regulatory proteins, perhaps established early in development, bind to specific gene promoters and recruit enzymes that modify chromatin, leading to this stable, long-term silencing that is passed on when the cell divides. So differentiation is about selectively silencing parts of the genome, putting away the instruction manuals the cell type will never need. Precisely. While a human cell might contain 25,000 protein-coding genes, a typical mature, differentiated cell might only be actively expressing, transcribing, and translating maybe 8,000 to 10,000 of those genes, the ones relevant to its specific job. The rest are kept silent.
Does the local environment, the neighboring cells, influence differentiation during development?
Absolutely. This is a crucial process called embryological induction. One group of embryonic cells produces signals that influence the developmental fate, the differentiation pathway of adjacent cells. A classic example is the cortomesoderm in early vertebrate embryos, which acts as a primary organizer, inducing the formation of the neural tube and other structures. Another is the developing eye cup growing out from the brain. Where it contacts the overlying surface ectoderm, it induces that ectoderm to thicken and differentiate into the lens of the eye. Much of embryonic development depends on this intricate cascade of inductive interactions between different cell populations. The exact signaling molecules and mechanisms are areas of intense research.
A complex interplay of internal genetic programs and external signaling cues. Another vital process for shaping tissues and maintaining health is programmed cell death, or apoptosis.
Apoptosis is essential. The body controls cell numbers not just by regulating the rate of cell division, but also by actively controlling the rate of cell death. Apoptosis is this genetically programmed, highly regulated process of cellular self-destruction. Cells initiate apoptosis when they are no longer needed, when they are damaged beyond repair, or when they pose a threat to the organism, like virus-infected cells or potentially cancerous cells.
How is this programmed death different from a cell just dying from an injury, which is called necrosis?
The difference is stark and crucial. Necrosis, typically caused by acute injury, like lack of oxygen or exposure to toxins, is a messy, uncontrolled process. The cell swells up, its membrane breaks down, and it spills its contents into the surrounding tissue. This leakage triggers inflammation and can damage neighboring cells. Apoptosis, in contrast, is an extremely orderly process. The cell actively shrinks, its chromatin condenses, its cytoskeleton collapses, and the nucleus breaks up. Importantly, the plasma membrane remains intact initially, but undergoes changes and displays signals on its surface. These signals attract specialized phagocytic cells, like macrophages, which quickly engulf the dying cell and its fragments before they can leak their contents. It's a clean disposal that prevents inflammation and collateral damage.
A tidy, pre-planned self-removal. What molecular machinery drives this process?
The core executioners of apoptosis are a family of proteolytic enzymes called caspases. These enzymes are synthesized and exist in the cell as inactive precursors, called procastases. Apoptosis is initiated when specific signaling pathways, triggered by either internal cues like DNA damage or external signals like death ligands, activate the first initiator caspases. These initiator caspases then cleave and activate downstream executioner caspases. This activation often occurs in a cascade, rapidly amplifying the signal. The executioner caspases then go on to systematically cleave hundreds of specific protein targets within the cell, breaking down the nuclear lamina, degrading cytoskeletal components, activating DNA-cutting enzymes leading to the characteristic morphological changes, and dismantling of the cell.
And this process is widely used in the body.
Extensively. Apoptosis is absolutely critical during embryonic development for sculpting tissues and organs, for example, removing the webbing between fingers and toes, or eliminating neurons that don't make proper connections. In adults, apoptosis is essential for maintaining tissue homeostasis. In tissues with high cell turnover, like the intestinal lining or the bone marrow, billions of cells die by apoptosis every single hour, precisely balanced by the production of new cells through mitosis. It's also crucial for eliminating damaged, infected, or potentially cancerous cells.
What happens if this apoptotic balance goes wrong?
A dysregulation of apoptosis is implicated in a wide range of diseases. Too little apoptosis cells surviving when they should die is a hallmark of cancer, allowing mutated cells to proliferate, and can contribute to autoimmune disorders, failure to eliminate self-reactive immune cells. Conversely, too much apoptosis excessive cell death contributes to tissue loss in conditions like neurodegenerative diseases, Alzheimer's, Parkinson's, heart attack, and stroke. Understanding and manipulating apoptosis pathways is a major focus for developing new therapies. For instance, designing drugs that induce apoptosis specifically in cancer cells.
That leads us perfectly into our final major topic, cancer, which you've described as fundamentally a disease of uncontrolled cell growth and division.
Exactly. At its core, cancer arises from the accumulation of mutations or other abnormal alterations in the genes that regulate the cell cycle, cell growth, DNA repair, and apoptosis.
The source material specifically mentions proto-oncogenes and oncogenes.
Yes. Proto-oncogenes are normal genes that play positive roles in cell growth and division. They might code for growth factors, receptors, or components of signaling pathways that tell the cell to divide. Think of them as the cell's normal accelerator pedal. An oncogene is a mutated or abnormally activated version of a proto-oncogene. This mutation essentially makes the accelerator pedal get stuck in the on position, leading to excessive stimulation of cell division, even in the absence of normal growth signals. Over 100 different oncogenes have been identified in various human cancers.
So oncogenes push the cell to divide. Are there also genes that normally stop division, like breaks?
Precisely. These are the anti-oncogenes, more commonly known as tumor suppressor genes. These genes normally act to restrain cell growth and division. They might encode proteins that inhibit progression through the cell cycle, or proteins involved in DNA damage repair, or proteins that trigger apoptosis if damage is too severe. They are the cell's brakes. If a tumor suppressor gene is lost or inactivated by mutation, the cell loses a crucial braking mechanism. This allows cells with other genetic damage, like activated oncogenes, to proliferate when they shouldn't. Often the development of cancer requires both the activation of oncogenes, stuck accelerator, and the inactivation of tumor suppressor genes, broken brakes.
It usually takes multiple hits failures in both acceleration and braking. But given that mutations happen, why don't we all inevitably get cancer? Why is it still relatively rare compared to the number of cells and divisions?
Thankfully, our bodies have multiple layers of defense against cancer development. 1. Many cells that acquire significant mutations simply have reduced survival fitness and die off, or are outcompeted by healthy cells. 2. Most mutated cells that do survive still retain some normal feedback controls that prevent runaway proliferation. 3. Critically, the immune system provides surveillance. Cells that start becoming cancerous often display abnormal proteins, tumor antigens, on their surface due to their mutations. Immune cells, like cytotoxic T lymphocytes and natural killer cells, can recognize these abnormal cells and destroy them before they form a detectable tumor. This immune surveillance is why people with suppressed immune systems have a significantly higher risk of developing certain cancers.
The immune system acts as a quality control inspector, removing defective cells.
It does. 4. And as we mentioned, it usually requires not just one mutation, but the accumulation of multiple critical mutations in different key genes, oncogenes, and tumor suppressor genes, within a single cell lineage to transform a normal cell into a fully malignant cancer cell capable of forming a tumor and metastasizing. This multi-step process takes time and is statistically improbable for any single cell, though the odds increase with age and exposure to mutagens.
So it's usually a series of unfortunate events, not just one bad break. What are the main factors that cause these initial mutations or genetic alterations?
Several factors can contribute to the DNA damage that leads to cancer-causing mutations. 1. Chance spontaneous errors. DNA replication and repair, while highly accurate, are not perfect. Random errors occur spontaneously during the copying of billions of DNA bases. Over a lifetime of cell divisions, these spontaneous mutations accumulate, and some may hit critical genes purely by chance. 2. Environmental mutagens. Exposure to certain agents significantly increases the mutation rate. A. Ionizing radiation. X-rays, gamma rays, radioactive substances, and even ultraviolet UV radiation from sunlight can directly damage DNA, causing breaks and mutations. B. Chemical carcinogens. A vast number of chemicals are known to cause cancer by damaging DNA or interfering with cell cycle control. Famous examples include components of tobacco smoke, responsible for a huge proportion of cancer deaths, especially lung cancer, asbestos, certain industrial chemicals like benzene, and aflatoxins produced by molds on food. C. Physical irritants. Chronic physical irritation or inflammation can increase cancer risk. For example, chronic abrasion in the digestive tract can lead to ongoing cell damage and regeneration. The increased rate of cell division required for repair inherently increases the probability of replication errors and mutations occurring.
So more cell turnover means more chances for mistakes.
Exactly. 3. Heredity. Some individuals inherit mutated versions of crucial genes, often tumor suppressor genes like BRCA12 or DNA repair genes, from their parents. This doesn't guarantee they will get cancer, but it significantly increases their predisposition because their cells start life already having one hit or one broken break, requiring fewer additional mutations to become cancerous. 4. Oncogenic viruses. Certain viruses are known to cause or contribute to human cancers. Examples include human papillomavirus, HPV, linked to cervical and other cancers, hepatitis B and C viruses, liver cancer, Epstein-Barr virus, some lymphomas, HIV increases risk of several cancers, partly via immune suppression, and others. These viruses can cause cancer through various mechanisms, such as inserting their own genes, which might be oncogenes, into the host DNA, integrating their DNA in a way that disrupts host tumor suppressor genes, or producing proteins that interfere with host cell cycle control proteins like p53 or Rb.
A wide range of causes, from pure chance to viruses. Once a cell does become cancerous, what are its key characteristics that make it so dangerous compared to a normal cell?
Cancer cells typically acquire a set of distinctive traits that enable their malignant behavior. 1. Ignoring growth limits. They lose sensitivity to normal signals that inhibit growth and division, like contact inhibition, and often don't require the usual external growth factors to proliferate. 2. Reduced adhesiveness. They often lose or alter the cell surface molecules that normally make cells stick together in tissues. This reduced adhesion allows them to break away from the primary tumor.
And that lack of stickiness is key to spreading, or metastasis.
Precisely. Metastasis, the spread of cancer cells from the original site to form secondary tumors in other parts of the body, is the deadliest aspect of most cancers. Because they are less adhesive and often more motile, cancer cells can invade surrounding tissues, enter the bloodstream or lymphatic vessels, travel to distant organs, and establish new colonies there. 3. Angiogenesis. As a tumor grows beyond a tiny size, it needs its own blood supply to deliver oxygen and nutrients and remove waste. Cancer cells often acquire the ability to secrete signaling molecules, like VEGF, that stimulate the growth of new blood vessels into the tumor. This angiogenesis is essential for tumor growth and metastasis.
So they break free, travel, and then induce the body to build them supply lines. Ultimately, why is cancer so often fatal?
Cancer kills through several mechanisms, primarily related to its uncontrolled growth and spread. 1. Nutrient competition. Cancer cells are metabolically highly active and proliferate relentlessly. A large tumor mass demands enormous amounts of nutrients and energy, effectively competing with and starving the body's normal healthy tissues. This can lead to wasting, cachexia, and organ failure due to lack of resources. 2. Organ disruption. As tumors grow, they physically invade, compress, and destroy the normal structure and function of vital organs. For example, lung cancer can block airways and destroy gas-exchanging tissue, impairing breathing. Brain tumors can increase pressure and damage critical neural centers. Liver metastases can destroy liver function. Eventually, the damage to one or more vital organs becomes incompatible with life. It's a profound disruption of the body's normal architecture and physiology.
A truly devastating consequence of the breakdown in the intricate cellular controls we've spent this time exploring.
It really highlights the importance of every layer of regulation we've discussed, from DNA repair and proofreading to cell cycle checkpoints, chromatin structure, feedback inhibition, apoptosis, all working together to normally prevent this kind of catastrophic failure.
We have covered an incredible amount of ground today. It feels like we've journeyed from the very essence of the genetic blueprint, that elegant double helix in the nucleus,
Through the complex machinery of transcription and translation, making RNA messengers and protein workers.
exploring the myriad ways cells control exactly which proteins are made and when through genetic and enzymatic regulation.
Witnessing the precise, tightly controlled dance of DNA replication and mitosis during cell division.
Understanding the necessity of programmed cell death, apoptosis for sculpting and maintaining tissues.
They're finally confronting how failures in these fundamental control systems can unleash the uncontrolled growth of cancer.
It really drives home how this genetic system isn't just abstract biology, it's the operational command center for literally everything happening inside you right now. It dictates daily health, influences aging, and underlies disease.
And understanding it at this fundamental level, I think, gives you a really powerful perspective on life itself.
And the complexity is just staggering when you think about it.
Yeah.
All those layers of control, the promoters, enhancers, insulators on the DNA, the feedback loops for enzymes, the checkpoints guarding the cell cycle, the proofreading systems fixing errors, the telomeres acting as division counters, the immune system patrolling for rogue cells. So many safeguards.
It's a testament to the robustness required for complex life to function. It also highlights the vulnerabilities when those systems fail.
As we wrap up this deep dive, maybe here's something to mull over. Think about that almost unimaginable precision, that incredibly complex choreography happening constantly, second by second, across trillions of cells in your body, all of them dividing, differentiating, communicating, functioning, all guided by this ancient molecular code stored in DNA. How much of what we perceive as our thoughts, our feelings, our very consciousness might simply be the emergent property of this unbelievably sophisticated molecular machinery faithfully executing its instructions?
That's a profound thought. And looking forward, as our ability to read, understand, and even edit this genetic code grows ever more powerful, what are the implications? For treating inherited diseases, for combating cancer and aging, perhaps even for influencing the future trajectory of human biology itself. The potential and the ethical considerations are immense.
Definitely food for thought long after we finish here.